Hello everyone, and thank you for attending today's webinar, Correlative Microscopy and OIM Analysis. Before we begin, I have a few housekeeping items. At the bottom of your console are several application widgets you can use. If you have any questions during the webcast, you can click on the Q&A widget and submit your question. We'll try to answer these during the webcast, but if a fuller answer is needed or we run out of time, you'll receive an answer later via email. A copy of today's slide deck and some links to additional materials are available in the resource list widget that looks like a green folder. You can expand your slide area by clicking on the maximize icon on the top right or by dragging the bottom right corner of the slide area. Finally, an on-demand version of the webcast will be available shortly after the live session, and it can be accessed using the same audience link that was sent to you earlier. Now, I would like to introduce Matt Nowell, who's our presenter for today's webinar. Matt is the EBSD product manager at EDAX, and he has a passion for EBSD and microstructural characterization. Matt joined TechSem Labs when he graduated from the University of Utah in 1995 with a degree in materials science and engineering. At TSL, he was part of the team that pioneered the development and commercialization of EBSD and OIM. After EDAX acquired TSL in 1999, he joined the applications group to help continue to develop EBSD as a technique and integrate structural information with chemical information collected using EDS. Within EDAX, Matt has been involved in a number of roles, including product management, business development, customer and technical support, engineering, and application support and development. In his spare time, Matt enjoys golf and pondering if changing the texture of his clubs will affect his final score. Now, over to Matt.
Thanks, Sue, for that nice introduction, and welcome everyone to today's webinar. As we get started, I'd first like to offer some acknowledgements. This work is on correlative microscopy, and so it brings in a number of other techniques. And to do this, it's required assistance from a number of other people. So we'd like to acknowledge Marty Crimp and his group at Michigan State, some folks at Kamika, Katie Rice, and Yingming Chang, Sanjeet and Ryan from Brooker from Nanoindentation, work with Ron Witt on Prius Analysis, with Suzuki-san from TSL in Japan for some TEM work, groups at NREL, University of Utah, and University of Toledo on some photovoltaic work, and then, of course, co-workers here at EDAX, Sean Wallace, Jens Rafelson, Tara Nilese, Renee DeClos, and Stuart Wright. We've all worked together to try to develop some of these correlative examples that I'll show you today. So I want to just start and talk just for a second about EBSD. I put this quote here from a talk at Microscopy and Microanalysis in 2005 from Austin Day, which is really, I'm biased because as I do this for a living, EBSD, probably the best measurement in the world. And so we kind of like that. And what's important about EBSD is it's an SEM-based technique that's automated, provides spatially specific orientation data, but also provides information about phase, grain boundaries, grains, and local misorientations. So you look at that and you say, what else would we ever need? That's a pretty full suite of data. The spatially specific part is important. If we think about what that means, it means we can make a map. We can see the data. A picture is worth 1,000 words. This is just an example from an aluminum extrusion where we see two different textures develop. These are orientation maps, so the colors correspond to orientations. And we can see the spatial distribution of the red and blue components are different between the center of this extrusion and the edge of the extrusion. So if we were just looking at the sum data or a chart of this data, we couldn't see these distributions as well as we can see it visually. It jumps out. So when we talk about correlating data, a lot of times what we're going to do is take one map from one technique and another map from another and line them up so we can start to see how the data from one compares to the data of the other. And so, the motivation for this, I really like this image. It came from a coworker, Jonathan, who took this on a nice vacation in Ireland. He saw this rock and took a picture, and if you look at the larger image on the right, you can see some grains there it looks like grains, and I think that's the important question. He said it looked like an OIM map. And so it kind of got us thinking of, how would you take a sample like this and correlate this image with an OIM map to see, is the contrast we see in one image telling us something compared to the contrast for another image. And so that's, just kind of a big picture example, fun example of why correlative microscopy is of interest. So thinking about correlative microscopy, traditionally it's talking about bringing light and electron microscopy together. But if we're going to talk about a wider scope of things, we're going to talk about different imaging and characterization modalities on a single sample and how we can bring together lots of different data approaches to bring more insight into the characterization of a given sample. The more contrast we have, the more information we have, and then the more we can learn about our material. And having this correlative data can also assist in characterizing material over a wider range of length scales, from centimeter or larger down to atomic scale, to really try to understand how the microstructure and nanostructure of a material is characterized, and then how we can correlate it with properties and performance of a material. So if we start with optical microscopy, this is an example from 1994. So it's something we've been doing for quite a while. This is one of our first covers from a journal. Of course, optical microscopy, generally to see the grain boundaries, you have to etch the sample, and not all the grain boundaries are equal. And that's a statement I'll make a few times during the presentation. And so as we etch a sample to try to reveal the grain boundaries, not all the boundaries etch at the same rates. So when we use this for correlative work here, our main point was to say EBSD would show us boundaries we can't see easily with optical microscopy, and so we can see twin boundaries better. And so this was an example looking at stress corrosion cracking in a material where we wanted to know what boundaries the crack actually propagates through, so we can put more of those boundaries in, or more of the boundaries that resist cracking in. And EBSD can directly find these misorientations through the orientation measurements that it makes. Thinking in terms of SEM images, SEMs now come with a number of different detectors. Usually they'll give us good contrast. So this is an example from a secondary electron detector and a backscattered electron detector. We see nice grain contrast. The thing to remember with EBSD, though, is generally we polish the sample pretty well, and then we tilt it. So as we tilt our sample to around 70 degrees, we're changing the path of the electrons coming out of the sample, so they're no longer going towards the traditional detectors, and so their contrasts are not as optimal. And so it can be more difficult to see. And I think that's an important issue because, when we think about these images, contrast is information. Contrast tells us what we want to see in the sample. And so for an example, I want to talk for a second about thin film photovoltaics. These are materials that are deposited on, and so they use just a few microns of material as the active absorber material. So they can be used, they use less material, so they can be lower cost and can be deposited different ways than traditional solar cells. And from an EBSD point of view, the reason this is interesting is that for silicon, the traditional solar cell single crystal devices were generally higher efficiencies than polycrystalline devices, but they were higher cost. But for some of the other thin film materials like CAD, TEL, or SIGs, polycrystalline devices have higher efficiencies. And so one of the questions being asked was why? And so if we look at different boundaries with a different technique, this one called Kelvin force probe microscopy, we can go into specific boundaries and measure the response of the boundary with this technique. And you can see at the voltage response on the bottom, the blue curve there, one grain boundary has similar performance. Grain boundary 1 has similar performance to the interior of the grain, where grain boundary 2 has a higher voltage response than the interior of the grain. And so again, these different boundaries have different responses, and we can go and measure the responses at specific grain boundaries with some techniques, and we can correlate that with the EBSD. Now, as part of that, we have to, of course, prepare a sample for EBSD. This is an example. This is a cadmium telluride sample that's been what's called cadmium chloride treated. And so the as deposited state is there on the left. It's pretty rough. And so to make it ready for EBSD work, we cut a surface in with a gallium focused ion beam. And so the glancing angle here is about 1.5 degrees. Afterwards, we get nice EBSD, as we see in the middle. And on the right, you can see the forward scatter detector orientation contrast image as we've cut into it. And what's interesting about this is you can see there's a change in the contrast from the bottom of the cut to the top of the cut. The microstructure changes. So if we think about what this kind of cut is doing, as we're cutting into this from a surface mill, we're actually sampling through as a function of depth in the material. So near where the beam first comes in, we're at the surface, and as we cut further in, we're going deeper into the material. So we're sort of depth sampling through this. And so what that means is if we look at this cadmium chloride treatment, this is what they call activation of the grain boundaries and makes it more efficient. If we look at it when it's first deposited, the film, we get the structure there on the left. It's very fine-grained. When they heat it up with this treatment, it's partially treated. We kind of stop halfway through. You can see we get a distinct bimodal grain structure with small grains on the bottom of the image and larger grains on the top. Those larger grains are occurring deeper into the material, and then when it's fully treated, all the grains have grown. So we can see that it looks like these grains are growing from the bottom of the layer up towards the surface through this depth information. And we can plot that out on a little chart here to say here's the grain size as a function of depth. And by knowing the grain size as a function of depth and sort of how it changes, we get an idea of what the growth mechanisms are. And so, once we understand what the mechanisms are, then we can start thinking about engineering for improved efficiencies. So we'll come back to this type of sample in a little bit, but I wanted to shift gears and we'll talk about a geological sample. This is a piece of material that has titanium and iron in for economic interests. And so it has an ilmenite phase and a hematite phase that is used for commercial extraction. And so the goal was to use EBSD to measure the grain size and shape of these constituent phases. So the reason this sample's interesting from a correlative point of view for a couple of different reasons. First, the hematite and the ilmenite have an epitaxial type arrangement. They have the same crystal orientation within the grains. So if we look at the patterns from the two different phases there, you can see the patterns look very similar. And the reason that is that the unit cells are very similar. They just have different atomic position occupancies within the cells. And that makes it difficult for EBSD alone to differentiate these consistently. You can see on the phase map there on the right, you see the hematite in blue, the ilmenite in red, and it's a very noisy looking phase map. And so what we're going to want to do is correlate the EBSD with some other technique to try to improve this phase differentiation. When we do it with EDS, we call this Kaiscan for chemical indexing scan. But before we get to that point, we'll just kind of review why we need Kaiscan. And so it's important to remember that EBSD, it matches to the crystallographic structure. So if the crystal lattices are similar, the EBSD patterns are going to look similar, and it's going to be difficult for EBSD to distinguish. So if the crystal structures are different, EBSD works very well for phase mapping. If they're similar, it has more of an issue, and then we get those noisy looking structures. So if they're not identically the same but slightly different, so in this case, this is an example from nickel and copper, which are both face-centered cubic. So the atomic occupancy, it fills the same spaces in the lattice just with different atoms, and the D spacings are a little different. So what that means is the bandwidth and the EBSD patterns are a little different. But the difference here is so small, at the full resolution of our highest resolution camera, it's only about 1 pixel. So it's pretty difficult to differentiate those consistently. And then if we think about going to lower pixel resolutions for faster mapping, the difference is even less than 1 pixel. So even in the best case, we need really high quality patterns to try to differentiate it just by the pattern, which is pretty difficult to do. And so generally with Kaiscan, what we try to do is use what's called we call it Kaiscan for chemical indexing, where we can use the EDS information that's collected simultaneously. And so by collecting it simultaneously, we're getting a direct correlation. So as we put the beam on a specific point, we get EBSD and we get EDS. we can use the EDS information to then drive the phase differentiation. So in this example here, we have 3 different phases. One's nickel, one's steel, and one's copper. So we can look at the spectra and decide, well, when it's iron-rich, it's obviously going to be the steel phase. When it's nickel-rich, it will be the nickel phase. And when it's copper-rich, it will be copper. We can define the phases by the chemistry and then determine the orientation via EBSD. So we get a full characterization of both structure, crystallographic structure, and chemical composition. So going back to this geological sample, this is just a little SEM image of the entire piece of a rock. So it's a low mag view of the sample, and then a low mag SEM image can be seen sort of kind of in the center of that rock. So we've kind of gone to the center, and we wanted to say, how can we get a good idea of what this sample looks like? So with the SEM, I'm using an FEI Tennioscope, and it has an application that allows me to map a large area of the sample. So you can see here, this is an image kind of stopping through the beginning of this. You can see the grids that show the number of different images I'm going to take. It started filling them in, and as it goes, it's going to put these together. It's going to stitch them back together. For any of this kind of correlation, we need good contrast at the overlaps to get good alignment. And when I played with this, when you don't have a lot of contrast, it's hard to tell if you're well aligned. It makes sense. But one of the things I wanted to point out with this is that in this application, we can collect multiple different SEM signals or different detectors. So we have different contrast mechanisms available to us to help us get this alignment. So if we look now through the end of this, we get a nice field of view of the different minerals that are present in this rock. And we can see the different contrast from the different detectors. So this is a backscatter detector there on the left. This is a low voltage SE detector on the right. And we can see that we get different contrast. So the bright grains in the backscatter detector on the left show up as dark grains on the right. So, you kind of have to know what the detectors are doing to try to visually interpret this qualitative contrast. But by having these different mechanisms available, we see the image in different ways, and that information can possibly tell us something. Our next step is how can we bring EDS into this? So we can take our map and we can define an area to do EDS mapping. And this is, again, a large field map here. With EDS, we then know exactly how the atomic number contrast we see with the backscattered images correspond directly to what those EDS elemental and phase signals are. And if we look at that here, now we can take our EDS data and we can now tilt the sample to 70 degrees so we can do EBSD and collect EDS at the same time. So we can collect EDS both flat, traditional, but when we tilt it to 70 degrees, we can also collect EDS data. We'll talk about some of the implications of that in just a few minutes. Really the big thing for something like this, it's kind of hard to see the scale bar, but this is, probably centimeter scale map. When we tilt such a large sample to 70 degrees and move it, you have to be a little more careful just being aware of where your sample is in the chamber. So I just want to make a note of that to be aware that your working distance and size of the samples becomes a little more important as you're at such high tilt angles. And so once we've done here, I can take an EDS EBSD map within the area of interest. We focused on these titanium iron-bearing grains here. And so now we have both EDS and EBSD information. And we can now look and say, let's go and look at the grain structure within the targeted ilmenite grain. So these are the small grains. So we're going to go and collect a data set at around 400 by 300 microns with a 600 nanometer step size in this little area here that shows up in the map. This is an interface of two different orientations within a phase cluster of the microstructure. And then we can now look within that area. This is the EBSD image quality map on the left. So you can see these striated structures where they're sort of running top to bottom on the top of that and sort of from the bottom left sort of upward to the right on the bottom. And within that area, we can see the titanium EDS map. in the middle and the titanium iron map in the right, and we can see the titanium and iron rich grains, which is going to tell us which phase is which, and then the EBSD will determine the crystal orientation. And again, the orientations for these are about the same. So if we look at this here, this is our original phase map on the left. It's detecting it mostly as hematite, but when we use that chemical information to determine the phases there in the middle with the Kaiscan phase map, we more accurately determine where the phases are located. But if we look at the orientations on the right, we can see that the two phases are epitaxial. They have the same orientation within these different areas, and then the orientation changes. It goes from one area to another. And that gives us some idea of how these minerals grew from solution. And if we look at this, we can see there are even smaller features observed in the microstructure. Now, like I mentioned, we're tilted to 70 degrees for this kind of a collection. We've positioned the detectors and designed the detectors so that they work at this condition. They work over a larger working distance range, and we can look at these type of large samples. But we do need to be aware that when we are tilted to 70 degrees, the shape of the X-ray interaction and creation volume has now changed. And so the absorption path length, the corrections used for that change a little bit than are used for traditional EDS analysis. So to help to account for that, I did a little study here where we compared how the chemistry is measured and quantified with the sample flat under normal EDS conditions, and then when we tilt it to 70 degrees, how much it changes. So I just used a basic root mean squared deviation to kind of get an idea for how much a compound will change from a flat analysis to a tilted analysis. And the first time we did this, I picked a couple of different minerals here, and you can see the values here, the 0 to 45, that's if we're tilting either directly to the EDS detector or if we have some sort of an azimuthal angle relative when we're tilted. That's not really an issue when the sample is flat. And so you can see for the chalcopyrite there, we're about, in general around 2% RMS difference. But with the iron oxide, at a 45-degree azimuthal angle, it jumps up to around 9 degrees. And so this was our initial high tilt quant. We weren't particularly impressed. So what we did is we went in and changed some of the quant models and things we did. So we improved our deconvolution through a Bayes statistical analysis. The background modeling was adjusted. The mass absorption parameters were updated. We modified the absorption correction for the high tilt and some of the fluorescence corrections that all should have improvements for the high tilt values. And sure enough, as we do this, our NUSAF here drops this down on the iron oxide sample from a 9% difference to about a 1% difference. And so we ran this down from a range of materials, and really everything, the worst case was again around that 1% difference. So we're really happy to say not only we can collect data, not only can we use data at high tilt, we can actually quantify it at high tilts with pretty good accuracy. Now, when we are tilted, there's some other issues to be aware of. Our interaction volumes are a little bit different. So our EBSD generates our data from a smaller volume than the X-ray data. So it's possible that when our EBSD is coming from one grain, our X-ray information is coming from another grain or another phase. But it is aligned within the tilt. And so the reason that that's important is, here's a little schematic. When we're shifted just vertically, we can put a correction in where we just space things and shifted a number of pixels along the direction of the tilt. And so this is just an example from a stack of different layers in a material. If we look at the as collected data, you can see the bright area of this element here doesn't line up with the film that we see in the image quality map, and that's just the difference. This becomes more important as the step size becomes smaller, because if you're looking at, micron or larger steps, then the interaction volume difference doesn't really show up. But when we're looking at submicron pixels, this interaction volume problem can be more highlighted. So we can shift these pixels now so that the data of the EDS lines up with the microstructure we see with the EBSD. And once it lines up, then we can use that for Kaiscan analysis on nanometer scale phase problems. And that's kind of why the reason I talk about that is because that's what we can go back to with these ilmenite grains, is that I can go closer with this and look at a smaller area with 100 nanometer step sizes to try to resolve some of these very fine features we see within these little striations. But again, we know that at the voltages we're using, we can go, when we're having these different phases within the same orientation, that we'll get chemistry coming from across the boundary. So in this little map here, we can see the shape of the interaction volume here, and there's this red line showing where there's a boundary at 15 kV. So we can see on the one on the left that we get chemistry across that boundary when our beam's positioned within 100 nanometers. And on the one on the right at 5 kV, we can keep the size of the interaction volume much smaller. And so by running at a smaller voltage, we reduce the size of the EDS interaction volume. I think that's pretty normal. The other thing to just note is that the EBSD sample tilt, it changes the shape of this distribution within the material. And so being able to model it helps us understand sort of when boundaries will be important. So what we can do is we can now take our same sample, go back to a flat sample, and collect the same data but at 5 kV. And so with our new Octane Elite detectors at 5 kV, they're pretty high efficient detectors at low voltages, a lot of low energy sensitivity. And so we can get still pretty fast maps under these conditions. And so there's no sample tilt, so there's no interaction volume anisotropy. So we're able to line up the data where we're putting the beam more directly, and that allows us to have a map that's more spatially fidelity for better correlation. And so this is just an EDS net intensity map. So that net intensity map removes background and overlap effects. We can deconvolute different elements. And then the important key, and one of the key takeaways for the presentation is we've put a function into OIM Analysis that allows us to correlate images from different techniques with the underlying EBSD software. So here you can see the EBSD map on the left. This is a Prius image. I'll talk about Prius in a few minutes. And the 5 kV elemental map on the right. And basically what we do is we go and click on different points that are coincident in the two different maps. And when we do this, there are a few different algorithms we can use for correlation. There's a direct correlation where we're just shifting one-on-one, a linear bivariate, and a quadratic bivariate. With all of these, there's a preview available. So you click some points, hit a preview, and then you can see how well that correlation is if you need to add or change some of the points. Another really neat feature of this is that the images can be correlated as a set. So I've only shown one elemental map here, but I, of course, have a whole stack of these available. So when I correlate with one, it applies the correlation to all those so they line up. And so once we do that, we're then able to say, here's our original EBSD phase map. We can take our 5 kV data and correlate it with the EBSD image quality, then use that to drive the Kaiscan phase map. And so what we're able to do now is resolve features with finer spatial resolution than we could at just our 20 kV EBSD analysis tilted. So we get better spatial fidelity with both the orientation and EDS chemical information. And once we have it correlated, then we can start treating it just like any other data stream within OIM Analysis. So here I've created just a histogram, applied a color to show it on the EBSD map. And then I can come draw, in this case I've drawn a little line going across the boundary. So I can see both the misorientations and the chemical difference as it jumps across both this larger boundary, but also these different epitaxial phases to see how the chemistry is changing from one to the other. So by correlating it, we can bring it all together. And when we think about this, we can correlate it with a number of other signals. So we can do this really with any spatially resolved or related image. So a few examples would be WDS, Prius, Electron Beam Induced Current imaging, or Cathodoluminescence CL imaging. And once it's in the software, we can use that to help drive phase differentiation we've seen here, but other correlative analysis. So just to give some examples, this is an example correlating WDS data. This is a sample that was a rock with unknown minerals. And so the matrix was identified as cobaltite. EDS showed bismuth, sulfur, and a little copper. But the what's colored green phase here was similar, but there was no copper present. And so what we did is we look for different candidate phases. So we take the measured chemistry, we go look through different crystallographic data. So in this case, it's the ICDD powder diffraction database. We look through the ICST crystal structure database, the American Mineralogist crystal structure database, looking for different structures that match the chemistry that we found, and then we try those structures against the pattern to see what matches. In this case, there weren't any that matched well. And so we said, well, we need some more information then. So we took this sample and we looked at it with now WDS, because if we look at the EDS spectra, here on the right here, you'll see there's a little hump where this arrow's pointing. That's what we call our HPD, or holographic peak deconvolution. That line doesn't actually match up with the spectra, and you can kind of see here the arrow by the bismuth. It doesn't match up, and so the question says, are we missing an element? So that drives us to say, can we resolve it now with WDS analysis? So if we do a WDS across that, we can now see there's some lead that's present in here that we didn't see originally with the EDS data. So if we add lead to this, our peak matching matches up much better. And now if we add lead to our searches, we find another of other candidate phases. In this case, we find a structure through the American Mineralogist for the lead bearing that's, I'm going to call it krupkatite. Sorry, I don't know how to pronounce those. And then one that's lilianite, that now we can actually do our EBSD pattern indexing once we have the correct structures. So now we can go and measure both the elemental information there on the left, correlate that all together to get the structural and phase maps on the right there for the EBSD. So WDS buys us some better elemental sensitivity and resolution, peak resolution within the spectra. The next thing I want to talk about is what we call our Prius imaging. And so Prius is a way of using the EBSD detector as an imaging signal detector. So the way I'm using it in this example is we've predefined 3 different areas on the phosphor screen, basically a bottom, a middle, and the top. We monitor the electron flux in those regions as we move the beam across the sample. So we're basically using it as a detector by taking a subset of the phosphor. And these different channels give us different contrasts. And like we saw earlier, more contrast and different contrast gives us different information. So if I look at our original sample with the Prius, in this case the bottom sample, Prius in the center, you can see how there's a little change in the contrast in there, but it gives us a little bit more detail in the center of that. And this was the signal actually used when correlating with the EDS information, and used the top signal, which will have the strongest atomic number contrast. And so generally it's becoming more common to look at these images, at least these 3 baseline images on all sorts of samples. So this is kind of a neat example from a thermal barrier coating on a nickel superalloy. So the bottom, we generally see more topographical contrast, like we see here. We see the roughness after preparation. In the middle, we see the orientation contrast. We see the grain structure there in the coating areas, and the nickel on the top is more single crystal. And on the right, we see the orientation or atomic number contrast, where we see the different phases that are developing in different regions of this coating. And so if we go within the nickel, I think this is just a really cool example, because we can actually go in and resolve the gamma prime microstructure within the superalloy itself. And so pretty much now when we're looking within our lab, on almost any sample now, we at least look at the different Prius Images to see the types of contrast we get available. So these are the 3 different images as the rows and 4 different samples as the columns. And you can just see how the different regions give different contrast from the same area. And the contrast is information and tells us different things that are going on from the electron interactions in the sample to give us different ideas of what phases, what grains, what topography, what strains might be present in the material. So we get a suite of complementary contrasted images with Prius. As an example, this is one of my most favorite examples. This is a sample that we're looking at some growth coatings. So we look at the SEM image. You can kind of see the different layers there, but the EBSD image quality map was a little dark. Not all the areas were going, and they wanted to try to identify the different layers and the different interfaces at the layers. And Ron, who was doing this, was asking, saying, how can I try to resolve these a little bit better? And my answer was to him, well, have you tried Prius? And he was like, no, I haven't tried it. I wasn't quite sure how to set it up. And I said, well, the beauty part of Prius is that it's automatic in the background. It just occurs while you're mapping, so we could just open his data set, pull up the results, and then see the different contrasts along the layers. So these are 3 different images, 3 different contrasts, and you can see how we see the different layer contrasts from different parts of the layer through the 3 images. And so you can even see some of the very fine layers that are occurring where 2 of the larger areas come together, and they were just wowed by this image. They said, this shows us exactly what we wanted to see, and it just happened as part of the regular EBSD mapping. So other techniques we can think about correlation. This is one that's ECI, or electron channeling contrast imaging. This is a technique where we're imaging the sample, and by positioning the tilt of a sample relative to the incident beam, we can get different channeling conditions. So you can see on the schematic on the right, when there's a small n, and is the variable there, we get channeling down through the columns of atoms. When it's a larger n, we get more blockage. So we get different channeling conditions and different imaging contrasts through the positioning of the sample. Again, I want to really acknowledge Marty Krimp here, who's an expert at this, and let me steal some of his slides to show it. But by positioning the beam, we can then take an image here where we can go to specific diffraction conditions. And when there's a defect present, like we see on the bottom right here, that's a little schematic of the lattice with a little dislocation line in there. When there's a defect in there, it interrupts the channeling conditions, so we can see dislocations with the ECI contrast imaging. And so in this case, we positioned it to see specific dislocations within a material. They show up in the ECI condition. And then we can think about doing what's termed HR, or high angular resolution EBSD, which is when we take a pattern, we compare it with a reference pattern or a strain-free pattern of the same orientation. We define different regions of interest. Those are the different blocks that are shown in the patterns there on the left. Within each block, we compare from one pattern to the other through a fast Fourier transform cross correlation. We then look at all these squares together, all the different correlations together, to find the shifts from one pattern to the other at subpixel levels. And from that information, we can then calculate a displacement tensor to get an idea of what the strains are from the reference pattern to the measurement pattern. And so if we do this type of work on the same sample, so correlating now the EBSD and the HR EBSD with the ECI, these are, with the ECI, we can find the defects related to a specific slip system. With HR EBSD, we can resolve the geometrically necessary dislocations along specific slip systems, and then we can go and start to compare the 2 images from the same areas to see how they represent the same deformation structures. Another technique where we can get some correlation is atom probe tomography. So this is work done in conjunction with CAMECA. And so with atom probe work, we have a very small, fine specimen tip where there's a voltage applied, so we can basically then laser pulse onto them to extract a few atoms from the tip surface. Those atoms are then projected onto a detector, and the position and the time of flight onto the detector allows us to determine both the composition and the original position on the tip. So we can get a reconstructed image like we see here on the right. We get basically atomic scale composition information. And so to think about why this is useful from a correlation with EBSD point of view, if we think about grain boundaries in a material, they can be defined several different ways. Generally, we think about misorientation angle, axis of misorientation if it's a CSL type. But we can also think about grain boundary composition and grain boundary segregation. So the natural question is to say, how can we target a specific boundary where we know the crystallographic characterization, but now we want to measure the composition? That's where correlating atom probe work with EBSD work is really of interest. So we can take a map like this where we've show us where specific twin boundaries are in a nickel superalloy. We can go in now and target a specific boundary, use a focused ion beam instrument to go make a lift out along the boundary, and then prepare a atom probe specimen along that specific boundary and do atom probe work. And so to do that, we need to put the boundary within about 100 nanometers of the tip apex, and then we can put it into the atom probe and extract the atoms. So this image here in the center kind of shows the probe tip. This is a transmission EBSD image where we see the boundary. So we know the orientation, we know the boundary geometry. And then on the right here now, we have the chemistry along that boundary. So we can see that there's more boron segregating to the boundary than there is carbon. And we can tie all this under multiple measurements to different misorientations, axis, and angles. So that's kind of a neat correlation. And then there's other correlations we can do with this as well, in that, we can get an EBSD pattern and we can get a transmission EBSD pattern from the tip. We actually can get these hit patterns that come off onto the position-sensitive detectors. And when you look at these, they look like EBSD patterns as well. So we can actually determine the orientation from the hit patterns, and we can correlate those together by keeping track of the geometry of basically the projection of the EBSD relative to the incident hit patterns. And by knowing the orientations, it helps reconstruct the tip geometry a little bit better. Other correlations can be made with TEM. Of course, TEM is another method where we're going to get higher spatial resolution than we get with regular SEM-based diffraction. This is just an example on the right or on the left. It's a TEM bright field image. You can see a triple point grain junction there in the center. The image B here is a transmission EBSD image quality map. Image C is a standard EBSD image quality map in the same boundary, and then D is the SEM image. So here we're correlating 4 different imaging modalities together. And when we do this, we can go in and look and say, on the left here, we're looking at the diffraction patterns we see in TEM, the small changes in orientation. On the right, we see those small boundaries showing up in the transmission EBSD work as well. And we can see that the measured misorientations correlate together. And then thinking of not only going to specific grains, we can think about going the other way and saying, let's look at a electron transparent area of the sample, looking at it in lower magnification. And we can see over that scale, so it's a scale bar in hundreds of microns, where we can get the grain maps of lots of different grains within a TEM specimen. And so this is important because for non-cubic materials, knowing which grains are going to have the correct diffraction conditions for imaging different defects can be important. So we can see a wide range of grain orientations with transmission EBSD to help guide TEM work on a sample. So it can be a much more efficient TEM specimen analysis time. So to do that, we can basically look and say, we know our EBSD pattern is here, and this would be a simulation of a measured orientation pattern, and we can identify a specific zone axis. So in this case, we'd be looking at the O bar 112, and we want to position this orientation in the center of the image to get the diffraction conditions we want. And so in this case, we would need to rotate from where the 0112 is. We need to go 10 degrees up and 7 degrees to the left. We'll position that. So assuming you have a stage that allows you to tilt the different angles, ranges, and that's what the circles on here show are ranges that would correspond to 5, 10, 15, and 30 degree tilts. We can then tilt our specimen to position to the correct diffraction conditions. And by knowing the ranges that are available on our stage, we then know which grains would be within range for diffraction imaging. And so we can set a tolerance angle of the map to say, show us all the grains within, say, 15 degrees of the normal, and those particular grains would then be colored and illuminated and can help drive the TEM analysis. So another type of correlation we can think about is correlating our orientation measurements with local mechanical response. So if you think about, we take our orientation data, and then we can think about, applying some sort of a load on it, and we know the material is going to deform, along relatively known ways. So we know a lot of times slip systems. We can kind of get an idea to say, how does that crystallography correlate and compare with a known physical force. So we've done this through Nanoindentation. This is some work that was done a few years ago. And so here, taking an orientation map, selecting different grains. So you can see the grains here in the orientation map are listed as G1 and G2. Their position on the stereographic triangle are then plotted. And as we choose those grains, we can measure the hardness with Nanoindentation. And so basically now we can go and plot hardness by orientation, and we do this for a number of grains and different indenter types, and we can start to see how we get good correlation of the hardness measurements with the crystallographic orientation, sort of as we'd expect, and the basal plane orientations in this case are harder than prismatic plane orientations. Which I think also generally makes sense. And so by knowing orientations and knowing mechanical response, we're able to put those together, and we can start to explore different aspects of the microstructure. So in this case, this is just showing some examples where varying the amount of load from a nanoindent within the interior of a grain. And so here we've used 5 millinewtons, 15 millinewtons, and 25 millinewton loads into different grains. In this case, it's a nickel superalloy. With the image quality map we see here, we can see some of the plastic deformation that's been induced. You can see some slip bands that have been induced. So by knowing the orientation and then thinking about, we can pick a grain, but we can also rotate a grain on the surface. We can think about positioning things relative to the slip system, kind of applying force relative to the slip system to be able to see how material will respond locally. And I think this is, I've seen work where people will pull out nanopillars to do some similar type work. But you can also see, we can see how grain boundary precipitates are visible, especially on that 25 millinewton load there on the right. You see some precipitates visible at boundaries. So we can think about visualizing how the plastic deformation field starts to interact both within the interior of a grain, but also as it approaches grain boundaries, and think about things like slip transmission. And so this map is what's called a grod map, or a grain reference orientation deviation map. So blue basically is there's no plastic deformation. As it gets up to the warmer yellow and red colors, we start to see more and more rotations within the grain. And so we can see from this that we see the direction of the deformation field that develops, as well as the strength of the deformation. And then if we think about positioning this near a grain boundary, we can start to see how this deformation field will interact with boundaries and precipitates, because we know different boundary types will have different slip transmission properties, and there are approaches for analyzing that sort of thing. Groups over at MPI in Germany have looked at and developed some work in this area that lets you see how the material responds on a local scale relative to the orientation and grain boundary behavior. And that kind of goes back to the idea I started with, is that, grain boundaries and EBSD go together hand in hand. Not all boundaries are equivalent. We can see this here in a few different measurements of grain boundary energy as a function of misorientation angle, as a function of specific special boundaries. And of all characterization techniques, EBSD is really the best tool for measuring grain boundary structure. One, it's automated. Two, it gives us a spatial range where we can measure lots of grains and lots of boundaries. And three, it's spatially specific and ready to correlate with other techniques. So for grain boundaries, really, EBSD is the best measurement of them all. And so kind of tying back up this concept to what we talked about originally, if we look again at grain boundaries in photovoltaic cells, we showed, though, that Kelvin force probe microscopy image where we said we can measure the voltage response of specific boundaries. Well, we can think about doing similar work where we correlate EBSD data with characterization data. So in this case, it's CL data, cathodoluminescence data. From the same area, we have EBSD data. And so within the EBSD data, there on the bottom, this is where we can identify boundaries as being random high-angle boundaries, or we can identify them as being CSL twin boundaries within the solar cell material. And we can compare the boundary types with the CL response. And basically, we can see that twin boundaries have basically the same electrical response as the interior of the grains. It's more of a flat line there if we look at the chart of the CL signal on the right. But the random boundaries become darker. They do not generate the same CL signal as the interior of the grains of the twin boundaries. So this is some really nice work by Abu Ras over in Germany, where it shows that different boundaries have different types, and we can correlate it with boundary types and performance. And so this now ties into some recent work done over in MPI. We're doing now 3D analysis with a FIB to get 3D orientation and grain boundary structure. Of course, 3D is also a type of correlation where we're correlating one slice to the other. But what's kind of cool about this is, on the top left image there, image A is an orientation map in 3D. Image B is a grain boundary map. But now in 3 dimensions, doing what we've done before is showing high-angle boundaries, random high-angle boundaries in map C, and the twin boundaries in 3 dimensions in map D, to differentiate random boundaries from twin boundary structures, because we know these boundaries have different performances for photovoltaic applications. And then because we have the 3D information, we can see that larger grains are developing towards the back contact, fine grains developing closer to the PN junction. So similar to what we inferred from a 2D cut in the material, it can be measured now directly in 3D, which then, of course, goes back and links things to saying, how do we know how these things grow when we deposit them, as we anneal them, how do the grains grow? Once we understand the growth mechanisms, then you can start thinking about how do you engineer the growth to improve your efficiency. So I kind of liked how that tied the story up together. So with that, I hope I've shown you, a range of ways we can correlate EBSD data with other imaging and also characterization techniques. I think we can say we've shown how samples that contain multiple phases can be identified phase mapped better and consistently more accurately by using other information, either EDS or Prius or WDS information, to help improve crystallographic differentiation. Bringing other information together allows us to build a better knowledge base. The simultaneous collection of EDS is standard, and that correlation routines have been built into the software allow us to bring in the images, directly correlate, and then apply features in OIM analysis for subsequent work. So with that, I'll stop and say thank you for your attention. I hope it was informative. And I'm going to bring up the question and answer window to see if there are any questions here that I can answer. So one question here is, where is the Kaiscan function in the software? To that, there are actually a couple of different places it's available. On the acquisition side, if you collect your data within Team, there's an option after you've collected the data to then apply the Kaiscan function within the Team software. So basically, once you've collected the data, if you double-clicked on it within your project tree, you'll then see an analysis tab that says Kaiscan. You click on that, and you have the option to now set up your Kaiscan a few different ways. I'll talk about that for just a second. Similarly, within OIM data collection, when you reload a data set, there's now an option to set up your Kaiscan. The big difference between those two is that Team uses the full spectrum available, where OIM data collection is only limited to the elements you selected during your mapping. You also have the option of doing your Kaiscan now in OIM Analysis. The advantage of doing it there is you have everything available you had in OIMDC and in Team, but you also now have the availability of using Kaiscan coupled with the partitioning functions in OIM Analysis. What that means is now you can do Kaiscan only where you need it. So if you want to do Kaiscan at a specific phase or specific pair of phases, you can target only those phases and leave your normal phases that don't require it alone. That's advantageous because it saves on reprocessing time with Kaiscan. Looking here for another next question down. It says, you mentioned different correlation functions. Is there one you recommend? So if I go back in my slides here a little bit. I think I can push this to the audience. So here, there's a direct, a linear, and a quadratic. I rarely use the direct because usually, because we're looking at a tilted image with EBSD, and then we often are looking at a flat image with other techniques, a direct correlation is pretty difficult. You get some skew due to sample tilts, and also the fact that, when we think a sample is tilted and flat, usually there's not really. So direct is usually not perfect. Linear and quadratic, basically, quadratic is probably the best, but you need a few more points. In this example, I think I usually use around 10 points at least. That's the one I generally use. I've gotten pretty good results with linear, but that's often because I'm using the sample in the same microscope, so I'm only changing the tilt, so there's not too much distortion. The linear works pretty well. As I brought in things from other techniques together, I generally try to use the quadratic because it gives me a little bit more flexibility, but you generally have to correlate a couple of more points along that line. But that's the one that I generally like. And so with that, I think I'll go ahead and stop. If there are questions that have been submitted that I haven't answered, I'll go ahead and we'll reply to those offline. So I thank you for your attention for today, and tune into next month's webinar. Thanks.